So good afternoon, everybody. I'd like to thank Josiane for inviting me, and also Fiat, who does a lot of work behind the scenes, and the OCC Organizing Committee for the kind invitation to come back to Paris, which is always a great pleasure. I'm going to talk to you today about stress adaptation, aging, and proteolysis, in particular the proteasome and the long protease, on which we've done some work over the years. We actually started looking at the question that I'm going to talk to you about today a number of years ago. This is a paper that a former student of mine wrote, Chikuchi and I wrote several years ago, back in 1990, published in 91, looking at the idea that free radical theory of aging really didn't quite work, and that perhaps we needed some more parts to that theory. At that time, ladies and gentlemen, we're having technical difficulties. Apparently they're in my pocket. So we were interested in the idea that systems for removing or repairing damaged proteins, lipids, or DNA might explain some of the involvement of free radicals in aging. And beyond that, we started looking at the ability of cells and organisms to adapt to stress, and that perhaps a reduction in ability to adapt or adaptational capacity might be involved in the aging process. And we've been working on that theory, this idea that adaptational capacity may not be strong in aging, for a number of years now, as well as working on a number of genes that are induced by adaptation, and as well, of course, as working on the proteasome and the long protease. So the idea, of course, that we've been publishing for 25 years now is the idea that proteins, once they're oxidatively modified, are rapidly degraded in the cytoplasm, the nucleus, and the endoplasmic reticulum by the proteasome, and that prevents their aggregating and forming inclusion bodies or becoming small peptides. And as I say, we've done a lot of work on this model over the years, and I'm not going to focus too much on the basics of it. But we can break this process down into a number of different steps now. So if we look at the overall increased degradation of oxidized proteins by proteasome, there are actually two major portions to this process. The first one is an immediate recognition of oxidized proteins and an immediate activation of existing proteasome to do a better job of degrading oxidized proteins. And by this I mean that nuclear 20S proteasome is activated by poly-ADP ribose polymerase, published this a number of years ago, and a new paper that's actually just out online, I think yesterday or today, at Free Radical Biology and Medicine, showing that the 26S proteasome is converted to 20S proteasome, the form that can degrade oxidized proteins better immediately upon oxidative stress, and I'll tell you a little bit more about that, but not much today. Secondly, there's this adaptational component, and this is when you have a system in which cells can adapt to the oxidative stress. If it's not overwhelming, then cells will adapt to that, and as I say, we've been very interested in adaptational capacity, especially with respect to aging. And there we see that 20S proteasome is synthesized, that PA2811S proteasome regulator is synthesized, that the PA200 regulator is synthesized. I'm not going to talk about that one today, and that the immunoproteasome is synthesized, and I will mention each of these today. So we find this two-step process, one immediate, one longer term, depending upon the level of stress. Now, I'm mentioning many different forms of proteasome. The core proteasome is this 20S enzyme complex here, and this is the one that degrades oxidized proteins very well. Another form that's very familiar to many of you is the 26S proteasome, and this has the core 20S proteasome, and at one end a 19S regulator, and there should be another 19S over here. This is the 26S proteasome. This is the form that can recognize and degrade ubiquitinylated proteins. So if you're looking at the ATP ubiquitin pathway, it goes through this form of the proteasome. There's also other forms of the proteasome. There's an immunoproteasome. There are other activators, including this 11S activator, also called PA28 alpha beta, or alpha, beta, gamma. There's a lot of different terminologies. So there are different activators, there are different forms, and there's also something called the immunoproteasome, and this is a form in which 3 of these beta subunits are exchanged for different subunits that rely on 3 different genes than the 20S proteasome, and you then form what's called an immunoproteasome, and its function has been argued about for years. And I'm going to come back to the immunoproteasome in a minute. So this is what we think occurs immediately following an oxidative stress, where 26S proteasome, this is the form that recognizes polyubiquitinated substrates, is converted to 20S proteasome, and that process seems to be helped by HSP70. This is the paper that's just come out today, and I won't show you the data for that this talk because it's not really much to do with aging. But what we find is that HSP70 binds to the 19S regulator, actually prevents it from being degraded during oxidative stress. There are a couple of other factors in here as well. Glutathione also plays a role, and there's another gene that's involved in helping this process occur, it seems. But in any case, 26S falls apart, gives you free 20S proteasome, which is then able immediately to degrade lots of oxidized proteins very well. And that's the key in this immediate response, is you make lots of 20S proteasome. Over a period of about 3 hours, the 26S proteasome reforms. Now this is horrendously complicated at first, but it actually isn't quite so bad. This is what I just showed you. This is HSP70 helping 26S proteasome. Here's the 20S with two 19S regulators making the whole 26S, falling apart, giving you 19S bound to HSP70 and free 20S proteasomes, this immediate response that enables you to degrade oxidized proteins very well. Over a period of 3 to 5 hours then, 26S proteasome reforms, and that's very important because there are lots of important cellular processes that rely on and require the ubiquitin proteasome pathway. And if you're going to adapt, you need to be able to do a lot of those. So that reformation of the 26S proteasome is important. But over the whole 24-hour period of an adaptive response, what you see is about twice the amount of 20S proteasome has been synthesized newly. There's these new orange balls, which are the immunoproteasome that's been formed. And there are also 11S regulators now bound to several of the 20S proteasomes, and some of these are immunoproteasomes. So this is the adaptational part of the response. Show you some data for some of that. So the activation of the nucleoproteasome, as I said, we published that several years ago, and this immediate response is shown here. Here's the increase in proteolytic capacity over a 24-hour period, responding to hydrogen peroxide. If you do the experiment in the presence of cycloheximide and prevent the induction of new proteasome, however, you get this burst of activity. This is the falling of the part of the 26S proteasome, freeing up 20S proteasome, but nothing more because you can't synthesize new 20S or immunoproteasome. As I say, I'm not going to have time to show you much more data on that, but this paper is out now and available online, in collaboration again with our old friend Tilman Grune in his lab. So the second part is this adaptation through altered gene expression, and the common experiment is one we've been doing for years. We first started doing this in 1989, and we're still doing it, where you have control cells, and cells are exposed to a high peroxide challenge, which causes some degree of cell challenge, a low peroxide pretreatment, and in the fourth case, a low peroxide pretreatment, wait for several hours, and then give the challenge. The experiments look something like this. This is in yeast. We've done it in everything from bacteria all the way through to human cells. These are the control cells. If you challenge them, you lose lots of them. If you peroxide pretreat with low dose, you get very little change. If you pretreat and challenge, it looks almost the same as control. In other words, they can adapt and they can survive. You don't see them being lost like this if you pretreat first. We see 30 to 40 genes upregulated during that adaptive response, and some of them are involved in proteolytic changes, and proteasome in particular, which I'm going to talk to you about today. So again, in this adaptive response, you see this initial burst of activity in the first hour. That's the falling apart of the 26S proteasome. And then afterwards, you get the further increase in activity that relies on de novo synthesis of proteasome and different proteasome forms. Now, the proteasome has 3 proteolytic activities, a chymotrypsin-like, a trypsin-like, and a peptidoglutamyl hydrolyzing activity, and all 3 of them are increased during this adaptational response, and all of them are inhibited by lactocystin and also by other proteasome inhibitors. I mentioned earlier that we've got many different forms of the proteasome, so which form of the proteasome are we actually seeing increase? Well, we think that 20S, as I said, is involved in degrading oxidized proteins, shown here. That 26S, whoops, there we go. That 26S, two 19S regulators plus the 20S, is involved in the ubiquitin pathway. It's key for that. And there's this other form, the immunoproteasome, with 3 beta substituted subunits plus 11S usually binding to immunoproteasome, whose function has been argued about for years. We proposed several years ago that it's involved in degrading oxidized proteins, but there's been little evidence for that. There's been a fair amount of indirect evidence, but nothing direct until recently. And so we think that the expression of all of these components, the 20S proteasome, immunoproteasome, and the PA28 or 11S regulator, are all significantly increased during adaptation. I'll show you some data for that. Here's this adaptive response. Here we're looking at various subunits of the 20S proteasome. So there's 4 different subunits we're looking at. They all increase. Here's the average of all 4. So there's about a doubling. Here's the subunit of the 19S part of the 26S proteasome. Absolutely no change in that. Here are the 3 subunits of the immunoproteasome. They increase even more. And most dramatically, the 11S proteasome, the alpha and beta subunits increase very dramatically, over 500% or even 600% increase. So all of these are going up. As I say, the 11S regulator most, then the immunoproteasome, and then the 20S proteasome. When we look at that here, this is proteolytic capacity of looking at the 28 alpha beta gamma knockout mouse. If 11S or PA28 were important in this adaptive response, you'd expect that the knockout mouse wouldn't be able to adapt quite as well, and that's exactly what you see. Here's the knockout. Everything else is fine, but because it can't induce PA28, it only gets a partial adaptive response. If we look at the immunoproteasome, this again is actually the first direct demonstration that immunoproteasome degrades oxidized proteins. Here's 20S proteasome from erythrocytes, immunoproteasome from spleen, where you get a lot of it naturally occurring. Here you can see that this only contains the beta 5 subunit. This one only contains the beta 5 immuno subunit. And the degradation of oxidized hemoglobin in both of them is very good compared to control hemoglobin. A little bit better demonstration of this is here, in which we've got proteasome and immunoproteasome isolated from the same cells, from meth cells, here induced by gamma interferon. And again, oxidized hemoglobin degraded by both 20S or immunoproteasome, oxidized ezrin degraded by both of them again. So we think that immunoproteasome plays a pretty important role, is certainly capable of degrading oxidized proteins, and we think it does it rather well, actually. Now, when we use siRNA against proteasome, immunoproteasome, or PA28, we find that siRNA blocks against all 3, blocks this induction of proteolytic activity. So all 3 are involved, and all 3 are specific. If we look at the ability of cells to adapt, a BRDU assay for DNA synthesis, or a cell count assay, if we use siRNA against beta 1I of the immunoproteasome, or PA28, in all cases, whatever we're looking at, we don't get a full adaptive response if we block any of these. And again, if we look at the protein carbonyl assay with an siRNA here against beta 1, we get lots more carbonyl. We don't get this adaptive response if we block it. So, excuse me. So, what's causing this induction? What's responsible for all of these subunits and different forms of the proteasome and its regulators for being induced? Well, a good guess was the NRF2 pathway. There were already some indications out there in the literature that NRF2 was involved in proteasome regulation, and my good friend Stathis Gonos has done a little bit of work on this. And what we see here is the induction of proteasome with increasing peroxide treatment. And here we're looking at NRF2, and you can see that here's a blow up in both cases. Here you can see the NRF2 is sort of punctated, it's all over the cell. When you induce with peroxide treatment, this adaptive treatment, you see that NRF2 now is localized in the nucleus. That's what it should do. It should move from the cytoplasm to the nucleus. You can see the localization here. Also shown here is the fact that with siRNA, we can block this increase in NRF2. There's both an increase in NRF2 and a mobilization to the nucleus. The increase is because you're not degrading it anymore, because you've lost the KEAP1 part of the pathway. What we show here is that with NRF2 siRNA, or knocking NRF2 down or knocking KEAP1 down, we can prevent part of this adaptive response. So the cell counts are not as good after peroxide treatment with an attempted adaptation. Here we see that we can block the proteolytic capacity increase, which is normal here, is blocked here by NRF2 siRNA or by retinoic acid, which also blocks NRF2. And we see over here that this is also true for hemoglobin degradation, oxidized hemoglobin. Now, is it all of the components of everything that I've shown you, are all of them responsive to NRF2 and the KEAP1 pathway? We think not. What we find is that 20S proteasome seems to be highly responsive to NRF2 knockdown and to NRF2 blockage. So is PA28. But if we start looking at immunoproteasome, we get mixed responses. We get a little bit of response it looks like initially, and then we lose it. When we look at the number of upstream regulators of the NRF2 pathway in the immunoproteasome, there's only one site. In the proteasome and in the PA28, there are several. So we're starting to look now at whether or not there's any actual regulation by the NRF2 pathway or by other pathways of immunoproteasome. And immunoproteasome may be separately regulated. But in terms of the proteasome and in terms of the 11S regulator, all of these inducers of NRF2 induce this improvement in cell survival and siRNA against NRF2 blocks. When we look at inducers of NRF2, 3 here, curcumin, sulforaphane, and lipoic acid, they all induce. And again, siRNA against NRF2 blocks all of those responses. So these are internally consistent for proteasome and an 11S regulator at the very least. Again, the mechanism we think is this induction of proteasome, 20S proteasome, immunoproteasome, and the 11S regulator. And immunoproteasome seems to be separately regulated from the others. So what happens in aging? Well, this is a slide from our old friend Earl Stabman, who was, along with many of us, Stathis and Bertrand Figuet, and others of us have been looking at the accumulation of oxidized proteins with aging. And here from several groups, this increase in oxidized protein with aging. And part of that, people have argued, may be increased production of oxidants. But part of it, certainly in our hands, seems to be a decrease in proteolytic capacity, and the proteasome seems to play an important role there. Lots of cell culture models suggest declining proteasome activity and diminished cellular capacity to degrade oxidized proteins with age. Tilman Gruner and our lab did several studies back several years ago looking at senescence models, and then another model in which we take cells that are capable of dividing, but which we grow to confluence and stop them dividing, and then we can grow them out for long periods of time so we can actually do aging in vitro, so to speak. And when we look at that, we find that there's a diminished proteasomal capacity. So here's senescence, a straightforward senescence model, and that decreases proteasome inducibility. So the studies that I've just shown you, looking at how you can induce proteasome during an adaptive response model, this is hormesis, are lost in late passage cells. Proteasome is no longer very inducible in these late passage cells. If we look at beta-1 here for proteasome, beta-1I immunoproteasome, or PA28 alpha, again in late passage cells, a tremendous loss of inducibility. If we look at the other model, this is now post-mitotic aging in vitro, so we're looking at cells that can no longer divide because they've been grown to confluence, they stop dividing, and then they can age in vitro. We can either speed that up by adding 40% oxygen, which we sometimes do. Here we see that they have a diminished proteasomal activity, and their ability, again, to induce proteasome is dramatically reduced in the older cells. So here's the old versus young, old versus young, old versus young for these 3 different subunits that we've been looking at a lot. So we think that proteasome adaptive capacity is compromised in aging and in senescence, and that may be playing some role in the aging process. How about mitochondria and the LON protease? Well, we've shown over the years, in particular Daniela Bota from our lab, the LON protease is particularly involved in degrading oxidized mitochondrial proteins, and Bertrand Friguet and Anne-Laure Boteau have done a lot of work in this area too over the years. And we find that LON now is strongly induced in mitochondria of young animals during all kinds of stresses: heat stress, oxidative stress, starvation. Here's a study here. Each of the different colors is a different concentration of peroxide. Here at 200 micromolar we get the best response, and each one of these is a different time point after a stress induction. You can see there's about a 7-fold inducibility of LON protein and of the activity, total activity in cells. If you look at the mRNA content, very small changes. We've now been able to show actually this is a translational activation of LON, not transcriptional. Heat stress, serum starvation, again, LON very inducible. The red bar here is LON. The black bar is HSP70, so you can see it's even better than HSP70 in terms of inducibility. It actually protects cells. Here's the increase in LON. 200 micromolar gives us the best increase. We can block it with siRNA. Over here, we're looking at carbonyls. If we use the same concentrations as over here, down here, where we're not getting much of a LON change, we get carbonyls produced. Here, where we get the best LON induction, we get very little carbonyl. There's no increase from control. And then it comes back down again, and we get lots more carbonyls. If we block, then with siRNA, then we get lots of carbonyls all the time, no matter how much peroxide we treat with. And again, the same is true of cell counts. The cells do much better if they're allowed to induce LON. If that's blocked with siRNA, then we get no cell protection. But this doesn't seem to occur in older cells or in senescent cells. So these are WI38s with going into senescence. Early passage, middle passage, late passage. Middle are fine. Much lower levels, less than half in the senescent cells. And here the level of LON, this is 2 measures of LON by JPDH, or LON to porin levels dramatically drop in the late passage cells. And at the same time, carbonyl levels rise significantly. And here's a study that should be coming out, we hope, soon in the Journal of Gerontology. I was quite amazed when we first managed to get this work, actually. This is similar to what I showed you a couple of slides ago, where in young cells we're able to induce LON. We get this very large LON induction, and in response with peroxide treatment, and in response to peroxide treatment, as LON is induced, we get no increase in carbonyls. Again, as I showed you before, if we use siRNA to block the LON increase, then carbonyls rise substantially, same exact experiment. These are middle passage cells, and here the LON induction is slow and sluggish. You don't get this rapid rise. You get a sluggish rise, but it eventually goes up. While it's rising, we start getting carbonyls, and then it's overcome when the carbonyls are brought back down again. You can see the carbonyls here. And these are the late passage cells. I just want to point out that here, what we did is because the LON levels were so low, we wanted to give a direct comparison. So we've actually repeated the early passage no peroxide treatment, the same protein back in this lane, just to give you a direct comparison. And these are the late passage cells. And you can see LON is dramatically down. Also, you notice that the levels of porin is a marker for mitochondrial protein. We're using a lot more mitochondria in this blot than we were here because there's so little LON. Basically, LON is just not inducible. It stays down all the time, even though it's the same model as here, and the carbonyl levels start high and just keep going up higher. So these senescent cells are just not able to mount a normal adaptive response in terms of LON induction. LON is an incredibly complicated protein. It has a lot of different functions, but its other main function, other than being a protease and degrading oxidized proteins during stress, normally in quiescent times, LON is bound to the mitochondrial DNA, and it's actually bound in the D loop of the mitochondrial genome, where it's absolutely required for mitochondrial biosynthesis. So anytime you induce mitochondrial biosynthesis or mitochondria being synthesized, if LON is not present in the D loop, you get nothing, and LON knockouts are completely non-viable. So what we're thinking is now that one of the things that occurs during stress is that LON comes off of the DNA, comes out of the D loop into the mitochondrial matrix, degrades oxidized proteins. This puts an immediate halt on mitochondrial biogenesis, and you can't have the two occurring at the same time. So there's a stress, non-stress role for LON that we think may be rather important in certain aging problems. We've got a paper that we're now in review, I hope, with Zsolt Radak, who's here somewhere, looking at LON in exercise, and what we're finding is that LON levels in aging, of course, decline. That's been shown by several groups now. But that in exercising animals, the muscle levels of LON don't go down as much, and there may even be some recovery in older animals that start exercising. So that's very encouraging in terms of LON being able to be recovered, and talks back to Malcolm Jackson's talk earlier of how LON may be inducible or may be recoverable by exercise in older animals. In overall sense, then, we think that adaptive responses to oxidative stress decrease in aging, an overall decrease in cellular adaptability. Proteasome, we think, plays an important role in this, mitochondrial LON protease, and other adapt and shock and stress genes we think are important in this overall senescing model of diminished inducibility or adaptability, I think play a role in aging. And for the data I've shown you today, all of the proteasome induction data came from a graduate student in the lab, Andrew Pickering, and all of the LON data came from a former graduate student, now a postdoc in the lab, Jenny Nago, and they and I thank you for your attention. Thank you very much. Gonna get started, yeah.
The paper is open for discussion, so we can start from here. Thank you, Kelvin. That was terrific. So, could you remind me what are in terms of the oxidized proteins, which of the amino acids serve as the recognition?
We don't think any of them do. So, I don't think anybody's had a good case for a particular amino acid that signals degradation. What we've sort of championed over the years is the idea that there's a partial denaturation that's sort of pretty much random depending upon where the amino acid hits occurred. But as long as it's minor, you get a partial unfolding. That partial unfolding exposes hydrophobic groups, which gives you an overall increase in surface hydrophobicity. And one of the things that the 20S proteasome loves are hydrophobic substrates. So they bind very well. Hydrophilic proteins are repelled by the proteasome normally. We've shown, along with Tillman over the years, that there's a number of different oxidized proteins for which early oxidation gives you an overall increase in surface hydrophobicity that correlates very, very well, one might say beautifully, with proteolytic susceptibility. That's a correlation. It's not proven. Nevertheless, that's the best correlation we've ever seen. And if you oxidize too much, then that goes away and proteins start actually binding together and forming aggregates, and then they cross-link and then they become lousy substrates. So to answer your question, I think it's exposure of hydrophobic patches that enables a protein to be degraded, and normally proteins don't expose hydrophobic patches.
So that means the different types of proteins are going to be drastically different in terms of how they are degraded in response to oxidative stress. Because not all have those same types of hydrophobic patches.
When you say drastically different in how they're degraded, I'm not sure.
Just in terms of the three-dimensional structures of the proteins, they have very, very different motifs.
Yeah.
Understand. And so what that would indicate is that there would be really a variety in terms of the way the proteins are recognized during the oxidation. If in fact it's that mechanism.
I'm not sure I follow. I think it doesn't mean that. I think it means that as long as a protein binds to the alpha subunits at the entry to proteasome, they can be degraded. So you can model it exactly, in fact, with heat denaturation. Take any protein, partially heat denature it, and you start making it a much better substrate, unless you go too far.
Okay, so all proteins are the same in terms of...
I think so. They all get... Because proteolysis by proteasome is all internal. Every protein has to go through the cylinder in order to be degraded.
Okay,
so I think it's binding the controls.
Let's move on to Giovanni's.
Kelvin, I was intrigued by a comment you made, and I may have misunderstood you, but you showed this data that if you used NR2 siRNA, the 20S proteasome could not be induced by hydroperoxide. You also then said if you used KEAP1 siRNA, you saw the same, but I didn't see the data. And I wanted to know how the two different siRNAs are working through a similar mechanism.
Yeah.
It
So,
doesn't make sense.
well, there are... So, KEAP1 is supposed to hold everything in the cytoplasm, right? And if KEAP1 is degraded, then then you can go to the nucleus and turn on the whole pathway. If you block keep 1, then there's nothing to, but keep 1 is also then protecting. It's not just binding, it's protecting. And if keep 1 is not there, then you get turnover. So what we're finding, and it depends very much, I didn't show you, I avoided it. It depends very much on how you time the assay. But if you time it, so to speak, just right, then if keep 1 is not there, you get no protection and there's nothing to go to the nucleus anymore. And that's the experiment in which it works. It's tricky, yes, I know. But that's the best we got.
So also an NRF2 related question?
Yeah.
Is this a general phenomenon that the NRF2 activation is impaired in aging?
So there's a lot of data about NRF2 and aging, and some of what I'm not showing you is we have data now, and I'm pretty sure Stathis has some data on this too. We have quite a lot of data now showing that NRF2 is decreasing, and the ability of NRF2 pathways to induce normal adaptive responses is diminishing. And at the same time, NRF1 is actually increasing in aging. And in some cases, NRF1 is antagonistic. Am I giving away your talk? Or? Okay, so we got the same data. And Stathis, and Bertrand, you got the same data too? Okay. So it looks like NRF2, I didn't show you this because it's not.
We'll have another talk for it.
Yeah. Well, these data of ours are not only not yet published, they're not even impressed, so I'm a little careful with them, but basically what we've got is that NRF1 looks like it's an antagonist to NRF2 that increases with age.
Okay, let's go on, please.
One simple question. In the repeating pathway, what is regulatory? The amount of proteasome or the E3 ligases?
What is limiting?
Limiting.
Normally or during oxidant stress?
No.
Normal. I think normally it's the ability of, it's the sequence of a protein that determines the rate at which it gets ubiquitinated. So this is Warshawski's idea. It's the NN rule, the idea that an inherent amino acid sequence that will determine the rate at which any protein gets ubiquitinated, that in turn determines how rapidly it gets degraded. Now, if you look at data from Alan Taylor's lab, then the E1 and E2 enzymes are the ubiquitin activating enzymes and ubiquitin initiating enzymes. Those are incredibly sensitive to oxidant stress and get shut down. I didn't show those data today because they're part of another talk, but they all get shut down for a good hour after oxidant stress, and then they get re-reduced. So that whole pathway, I think, is shut down and then reactivates about 3 hours afterwards, after the stress is gone.
Okay, quick question from
Kelvin, do you have any data about the quality of the proteolytic activity with age, about the type, number of peptides which are produced? And second, for example, there is also a protein splicing which can occur within the chamber of the proteasome. And does this change with age?
Yeah, we don't have any particular data on that ourselves. Part of the answer to your question may be the immunoproteasome. As you know, the immunoproteasome produces slightly longer peptides than the 20S or the 26S proteasome. And those slightly longer peptides are the ones that will fit into the cassette for the MHC class 1 pathway. And that's why it's been called the immunoproteasome, because of the linkage with self-recognition on the cell surface. We actually proposed something we called the proxy hypothesis several years ago, which is the idea that immunoproteasome should be able to recognize any oxidized protein in the cell, which means it's a simple way that adventitious oxidation can give you a portion of every protein on the cell surface through immunoproteasome, provided immunoproteasome could actually recognize an oxidized protein, and now we've demonstrated that. So we think we're going to go back to that hypothesis again. How it changes in aging is not yet clear.
Okay, we still have time for one last question. Sorry?
Thank you. Excellent. In case of young age, there is still a very defective amount of oxidized protein. Is this a reason of the failure of the proteasome system, or oxidized proteins might have some physiological function?
In old age or
young age?
In young animals? I think like anything else, there's always going to be some level of oxidized protein around. How much is going to depend on the rate of production versus rate of removal. I don't have any I mean, I think that a physiological function is that if you've got a piece of every protein from going through the immunoproteasome, through the MHC class 1 pathway, then you've got a simple way for cell recognition. So that may be one physiological function.
Okay, thank you.